Hello, my name is Ryan White, and my presentation today is over drug-resistant bacteria and our efforts to fight it utilizing anthraquinones derived from natural sources. Drug-resistant bacteria and viruses are one of the most dangerous developments in recent medical history. Antibiotics have saved millions of lives, but as pathogens evolve, they become immune to many of our most effective medicine. Our project aims to fight back, providing effective and localized treatment of infections and illness without the possibility of developing bacterial resistance due to our unique, light-based mechanism of action. We discovered that Japanese knotweed, rhubarb root, and yellow dock extracts are extremely promising out of the panel of plants we've investigated so far. Medicine and natural products have a long history together. Penicillin, for example, is one of the most important discoveries in human history and is derived from a natural source. Some plants make secondary metabolites that can be activated with light as a part of a clever defense strategy. These natural photosynthesizers absorb light to facilitate photochemical reactions that are toxic to predators, such as in the example of giant hogweed. Imodin is a photosensitizer that has antimicrobial properties when exposed to light. Photon absorption produces an excited state that creates deadly reactive oxygen species that can wipe out bacteria upon contact. The most important of these species to our research is singlet oxygen. Extracts from plants such as Japanese knotweed, rhubarb, and yellow dock have been reported to exhibit antimicrobial activity. These also contain various anthraquinones, which are known to have photosensitizing properties. We believe that by utilizing the antimicrobial properties of imodin and other anthraquinones, we can counter the epidemic of drug-resistant pathogens. Our light-based treatment overwhelms pathogens with reactive oxygen species, preventing them from evolving further immunity. Our goal is to compare the photo-antimicrobial effects of these root extracts. We used several different extraction methods and fractionation techniques for this comparison. 5 grams of each sample was extracted with 200 milliliters of ethanol at room temperature for 24 hours. The ethanol solutions were filtered and left open in the air to evaporate at room temperature. We repeated the extractions under identical conditions, this time, however, fractionating the extracts between water and dichloromethane. Around 5 grams of plant material was used in each extraction, and the recovery percentage is listed. Extracts were prepared as stock solutions in DMSO and tested in an agar diffusion assay against S. aureus under dark and light conditions. And as you can see, all of our extracts show antimicrobial activity under light conditions, with average inhibition zones around 1.5 centimeters. Japanese knotweed, rhubarb, and yellow dock root extracts all show strong inhibition zones. DCM fractions generally show stronger inhibition than aqueous fractions and are potentially stronger than simple ethanol soaks. However, more tests on this are needed. We also conducted a series of TLCs, which show very high mobility in 10% methanol and DCM solutions. In the future, absorption profiles of the extracts are being analyzed to observe optimal wavelengths to use in subsequent photo-antimicrobial assays towards the optimization of the activity. Yellow dock extract, for example, can be activated by blue, green, or red light, while Japanese knotweed extract is likely best activated by blue light. We also have previous studies to look back on, reporting on the antimicrobial properties of yellow dock. However, they did not investigate its photo-antimicrobial properties. Furthermore, our studies show no antimicrobial property in the dark, which contrasts with the published study. We aim to carry out a systematic investigation for comparison and to follow up on the article. I'd like to say thank you to the UTA Department of Chemistry and Biochemistry, to Dr. McFarland, and the rest of the lab. Thank you.